then. Hi, it's so great to meet you. Welcome to our podcast, I guess.
Thank you. It's great to meet you guys as well. I've never done anything like this, so pretty
Oh
excited.
yeah, definitely. This is new to us too. I think this is our second time we've had a professional kind of come in and talk to us for this type of thing.
Oh, awesome. Well, that's a
Yeah,
super thing to do, so I applaud you guys.
thank you. Yeah. So I'll just start with a few basic questions, just to kind of warm us up. Um, how about you introduce yourself and just kind of give us a good idea of who you are for the viewers.
Okay, sounds good. Sorry, you're gonna hear my cat causing a
No, you're
tremendous.
fine. You're good.
But my name is Laura McAuliffe. I'm originally from Boston, Massachusetts, but I moved to Charlottesville, Virginia when I was about 13 years old. So I went to high school there, and then I went to my undergrad at Christopher Newport University, where I got my Bachelor of Science in Psychology. And then after that, I worked at the University of Virginia's medical school for a little bit during my gap year before my PhD, where I did some clinical research assistance with type 1 diabetes trials there. And then after that was done, I began my PhD, which is where I'm currently at and in the middle of now at George Mason University. And I'm currently studying broadly learning and memory and how these processes develop first, and simply how some aspects of those things work in the brain on both a molecular and a cellular level. So, brief introduction to what I do. But also some general things. I always grew up playing, different instruments, and I was a snowboarder. I currently mostly am like rock climbing, but with kind of all these different things I've dipped my toes into, they never really clicked as the thing that I wanted to do with my life and for my whole life until I started studying psychology.
That's awesome. And yeah, what instruments have you played?
So I mostly played the violin. I started that when I was like 4 years old. And
Oh, same.
then,
I played the violin too.
oh, no way. Oh,
Yeah.
awesome. Classical?
I do it through my high school. But I've done it since middle school. But it's been more just through my class and stuff like that.
Oh, awesome. That's a good one to learn for sure.
Yeah, I think it's awesome.
Yeah.
Um, okay then. Let me see. Um, so you said you were working towards your PhD, correct?
Yeah, I just finished my third year and I'm going into my fourth right
That's
now.
awesome. So you're going to be getting your PhD soon.
Yeah, someday. The thing that they don't really tell you is that there really is no distinct timeline for these things. It's really mostly
Right.
just your projects and how your experiments are going and all of that. So, relatively soon, I like to think, but I'm along for the ride, so we'll see.
Right. Yeah. I totally understand that.
Yeah.
Um, how has that journey been?
So, it's been quite the interesting journey. Um, when I was applying for PhDs, it was when I was in this phase of like, okay, I really know what it is I want to do after studying psychology and a little bit of neuroscience in undergrad. But do I have, enough research experience and everything to actually get there? So the process of applying was pretty long and pretty stressful, but once I got into the lab that I am currently in now, I started and realized that, wow, I actually do really like this. And the lab that I'm in now was a little bit of a different road than I was used to, at least compared to like my undergrad research. I was very heavy in psychology, and I did a lot of research in like executive dysfunction and cognitive flexibility and autism spectrum disorder, and then educational psychology. So I got into this lab because when I was an undergrad, I also took a real interest in the biology of the brain and neuroscience. And again, this lab that I'm in is pretty heavily based in cellular and molecular neuroscience. So I decided to take the chance and come here and do my PhD in this lab, and realized again that, wow, I do really like this side of psychology and what I'm studying. So overall, I find it extremely easy to find good things in every day, even when I'm running in to do experiments for hours on end, or if my experiments aren't working, sometimes it's just very, very easy to remember why I'm here and why I'm enjoying it. But the PhD has been a lot of lessons as well. Um, it's been a lot of like focusing and staying focused even when it's hard. I kind of mentioned troubleshooting experiments and experiments not working. When you're doing like a psychology or neuroscience based program, depending on what you're doing, that's actually pretty common. Is that you have to spend a lot of time fine tuning experiments, and you'll go in and you'll test one thing one day, won't work, and it's the same thing for days or weeks or months on end. So it's been a huge lesson in resilience for sure. But again, I absolutely love what I'm doing, and I love the brain, and my PhD has done nothing but further kind of confirm that this is exactly where I want to be and what I want to be doing. So, yeah.
Oh, that's awesome. And I'm also doing research in neuropharmacology, and I noticed in your bio that you do research in molecular neuropharmacology. So I was just wondering, what sort of experimental methods do you use to run your experiments and stuff like that?
Yeah, so that's a great question. Neuropharmacology is actually what really got me into neuroscience when I was an undergrad. Because as I mentioned, I was a psychology major, but what really kind of channeled my interest in neuroscience, or started my interest in neuroscience, was when I started getting into and reading about how drugs are working in the brain, and just all these weird little things, like the fact that you can develop some sort of molecule that represents some sort of endogenous chemical that acts as a neurotransmitter, and that drug that you just developed that's molecularly similar to that neurotransmitter will go and act on those same receptors, was absolutely fascinating to me. So I went down rabbit hole after rabbit hole after rabbit hole on neuropharmacology, and that's how I kind of developed a pretty good basis in biology before getting into my PhD in neuroscience. So, in terms of what I'm doing with neuropharmacology now, I also took an interest in the kind of currently booming idea of psychedelics being used for therapeutic reasons for people with PTSD or depression. And I'm writing a paper right now that is basically talking about the molecular mechanisms of which ketamine works to kind of alleviate the symptoms of depression in people who have tried getting treatment but haven't had any kind of successful remission with typical antidepressants like SSRIs. So that paper is, again, it's a review paper, so I'm not doing anything hands-on with ketamine, at least not right now, but I've done a lot of research on kind of how exactly it's working in the brain on the level of different receptors, and how it's working on the level of different receptors causes you to feel the way that you feel when you're depressed, or causes you to kind of present with the memory deficits that you get when you have major depressive disorder. So stuff like that for that project. And then in my current project right now, what's pretty cool and something that you don't automatically think about is that when you're doing neuroscience research, sometimes when you're using some sort of pharmacological agent, the only, or, the way you use it is not just to study exactly how that drug is affecting the brain. You can also, when you know how it's affecting the brain, use it as a tool to study something else, which is what I'm doing. So right now I'm running an experiment that uses a method called in vitro electrophysiology, which is a way of basically just recording the electrical activity of neurons in the brain, following different types of electrical manipulation. And the receptors that I study are called NMDA receptors, N-methyl D-aspartate receptors. And if you haven't learned about these receptors broadly, they're kind of referred to as our plasticity receptors. So you hear about neuroplasticity, how the brain might change itself as a result of certain environmental experiences, or how it might make itself more efficient in some ways. Well, you have the level that you can think about it in terms of just the brain is very plastic. And then you can go one more level down and you can say, well, different networks within the brain have kind of different levels and different ways of going about this plasticity. And then you can go zoom in another level and you can say, well, the things that are driving these specific changes in plasticity are actually happening on the level of individual cells as well. And you zoom in and you find that there are these weird little groups of receptors that belong to certain receptor families and are associated with very specific neurotransmitter groups that are playing into this cellular plasticity that leads up to this kind of general plasticity that we think about in terms of just how the brain is. So what you can do is you can target a field of neurons or different cells, or maybe even a pathway in the brain that expresses a lot of these kind of plasticity controlling receptors. And again, you can electrically manipulate them with in vitro electrophysiology, and you can look at what happens after certain manipulations. Well, since I'm focusing on a certain drug, a kind of big approach in certain experiments like this is to see, well, what happens if I block this receptor instead of activating it? So now you're looking at, okay, what is this receptor doing when it's activating? And then you can also look at, well, what's happening when this receptor is blocked? And you can use drugs to do that. There's this drug that is called MK801, which basically goes and it sits in the ion channel pore of NMDA receptors and prevents their activation. And MK801 is a chemical cousin of ketamine and phencyclidine. And what's interesting about these NMDA receptors is that they have what's actually called a ketamine or PCP binding site. So these are the receptors that different drugs like ketamine actually specifically go in and target. So just to bring it kind of full circle, even though right now I'm not doing any experiments directly with like ketamine or anything like that, I'm using kind of chemically similar agents as a tool to look at what these receptors are doing differently in different environments. But again, you think about that, okay, well, this receptor has this specific binding place for ketamine. Well, cool. Then we can think about, okay, well, maybe this is how ketamine is playing into the alleviation of depression symptoms that we've seen in pharmacological trials. So a lot of interesting stuff there and probably a little bit of rambling. I could talk about this stuff for forever, but I'm going to pull myself back in and let you guys ask the same question that answers that.
That's very interesting. So I was wondering, so I know memantine is a clinically, like a FDA approved drug that's used like clinically for neurological and psychiatric disorders. I was wondering if you've ever used that for your experiments to like block the NMDA channel.
I haven't used that one specifically. Um, Mostly, I think, actually, no, I'm not sure about the specific kind of regulations around memantine, but I know with ketamine, even though it's used in clinical trials, it's kind of hard to go through the process of like getting those even to just use as tools in your research. So the reason why I'm using MK801 is because it's pretty easy to get for just laboratory research. And then also, it's kind of the drug that's been used the most for the reason why I'm using it. So a lot of times when you're designing an experiment or trying to figure out what the best way to go about it is, when there is kind of this tried and true method of doing something, a lot of times if that method is not your kind of direct thing that you're trying to assess and you're using it for something else, then you want to go with what the previous literature says. So I haven't used memantine in any of my research. And that's honestly probably the one I've heard or learned about the least out of all of kind of like the chemically similar compounds. But that's a good question.
Okay, so the next thing I kind of wanted to ask, so like ketamine and like all these other like drugs, like many of them are like antagonists, and I was wondering like how can we kind of make sure like they don't impair like like tasks that kind of use like hippocampus or like amygdaloid functions as much when we take them?
Yeah, so that's a great question. So, one of the main things that you will see when you are looking at or reading through the research on ketamine is the word acute ketamine. So, when we think of the word ketamine, ketamine along with all of these other psychedelics are pretty heavily stigmatized. So, when we think of ketamine, we think of it as this, really intense dissociative that people take and just kind of it kind of just makes it much harder to function normally when you're on this drug because we typically hear it as this really intense thing to do. And that's true, and that will happen when you take kind of bigger doses of ketamine. It's also used as an anesthetic, so if you take a lot of it, then you're kind of just knocked out. But again, the word that you'll see a lot in the ketamine literature is the word acute. And this means that it's not necessarily a microdose, but it's a pretty small dose of ketamine that you administer, and it's enough to kind of evoke these benefits that we've seen, or at least the benefits we've seen on a cellular and molecular level in animal trials, in humans, kind of like on a behavioral level in terms of they are feeling better, in terms of their depression symptoms and all of this, but they're not necessarily inhibited completely from, the world around them. A lot of people will report that after getting these kind of clinical acute administrations of ketamine, that even though they do feel kind of altered, or they feel like they have this like, trippy feeling going on during the session, and sometimes it does feel a little bit stressful. A lot of times people will report that that feeling quickly goes away after the trial, and a lot of times they'll report that they do feel some alleviation of depression symptoms that lasts for quite a bit longer after that. So people have done some studies on that, and they've looked at how people were affected after these kind of very small dosages of ketamine. And, to my knowledge and from what I've read, it seems that with this dose that, we're using in clinical trials, maybe at the NIH or wherever else they're testing this, that it doesn't seem to really inhibit people's memory or functioning after the trial too much. So again, while people will maybe feel altered in a way while they're receiving the ketamine treatment, it's not a big enough dose to make them fall asleep or go unconscious as if they were taking an anesthetic dose. And again, from what I've read, there have been kind of reports on the behavior and how people have felt after. And I don't think there's been a huge report of people losing memory functions or anything like that. Because an important thing to realize in situations like this is that even though we kind of think of NMDA receptors, for example, as plasticity receptors, so we typically will tend to think of something like that, as this unidirectional thing, especially when it has kind of more of a positive valence to it. So, okay, you activate your plasticity receptors, you increase neuroplasticity, which means you increase the strength of, certain brain functions. It's got to be a good thing. Well, what's really important in neuroscience is that you have to remember that sometimes too much of a good thing can turn into a bad thing, or even with these NMDA receptors, these plasticity receptors on the cellular level, when we're talking about kind of an increase in brain function or the strength of a pathway, on the cellular level, we refer to that as LTP, or long-term potentiation. And that on the cellular level is a strengthening of that synapse, which is allowing it to more effectively communicate with other synapses. But what can also happen, opposite of LTP, is you get what's called LTD, or long-term depression. And it's literally the opposite of LTP. LTD is this functional weakening of brain cells, and NMDA receptors are in charge of that as well. So when you're working with some sort of drug like ketamine, and it's targeting this receptor like the NMDA receptor that we know is in charge of plasticity, if ketamine, since we know it's an antagonist, is blocking that receptor, it's not necessarily fully a bad thing, because these receptors are a lot more nuanced than kind of they turn out to be, which is true with a lot of things in neuroscience, is that, there's a lot of these receptors we think as, doing something quote unquote good in our brain, but if you change how that's working, sometimes it can turn into a bad thing. So when you block these NMDA receptors with ketamine, it's not necessarily that you're blocking the hippocampus from being able to do a bunch of its positive functions. You're actually targeting these NMDA receptors in a way that makes it so that these receptors are less likely to kind of induce a type of plasticity, long-term depression, that causes this weakening of synapses, which leads to, or we at least think it leads to, deficits in learning and memory in people that have major depressive disorder, for example.
Okay, so the next thing on the topic of like NMDA receptors, in terms of like you talked earlier about like sort of like your research on turning them on and off. So I was wondering how we could sort of pharmacologically like I wouldn't say prevent magnesium from binding, but sometimes like magnesium can like bind to these receptors and kind of like stop like calcium ions from coming in. So like I was wondering how we could like regulate that.
Oh, that's a really good question. So what's interesting about NMDA receptors is that magnesium isn't necessarily like in this certain situation going in and binding to NMDA receptors. It's actually the case that when these NMDA receptors are not activated, that's when they're blocked by magnesium. So you have kind of your ion channel where the calcium is actually flowing in through these NMDA receptors. And that's where the magnesium is sitting when these NMDA receptors aren't activated. So magnesium isn't necessarily playing this like activity dependent role of inhibition of NMDA receptors. It's kind of preventing them instead from becoming overactivated when they don't need to be. So with that being said, certain types of activation can lead to LTD or LTP. So that calcium influx that you talked about, when the magnesium is sitting in that ion channel, calcium can't move into the cell. But when you have a bunch of different kind of coincident events that happen at the synapse that causes the magnesium ion to become dislodged, once that magnesium ion becomes dislodged, that's when you get, again, this kind of activity-dependent influx of calcium into the cell, which can result in a calcium-dependent activation of certain proteins or kinases that are sitting inside the cell that are associated with learning and memory, for example. But to bring it back to long-term depression and long-term potentiation, it was initially thought that long-term depression was caused by this kind of low, slow influx of calcium after some sort of activation, whereas long-term potentiation was instead caused by this high and fast influx of calcium, which makes it all the more harder to study, because it's not as black and white as, a bunch of calcium coming in is bad and leads to LTP, so we need to block it. Or LTD, so we block it and we prevent that LTD. It again is like, a little bit more than that. It's instead based off of, the level of calcium that's coming in, if any, at, this sort of synapse or during this sort of activation. So yeah, it gets kind of hard to think about how we go about studying that stuff and figuring out the pathways, but there have been a lot of people who have kind of done research on this and figured out a lot, especially within the last decade, that have provided a new foundation for studying how this stuff works, because the NMDA receptor is quite a tricky one, especially just given its involvement in LTP and LTD and how these processes are kind of semi-ubiquitous throughout the brain. It gets harder and harder to figure out. So again, it's not necessarily just about exactly what the magnesium is doing. So it's not the case where it's like, okay, we put a magnesium ion in the channel pore, we're blocking something bad or we're causing something good. It's more so what is happening, or what are all the different things that are happening that are leading to all of these different kind of results that are happening when the magnesium comes out of the ion channel. And that's what's interesting about some of these kind of pharmacological agents, is that with MK801, for example, and ketamine works similarly, it's this kind of activity-dependent drug, at least in the situations where I'm using them, where you have to, in order for it to kind of get into the channel and kind of sit in there, it's basically doing the same thing as magnesium. So you administer it and, it does its whole kind of chain of events, and then it kind of gets that spot. And again, is doing that same thing. So we use that as this kind of magnesium ion to look at the downstream effects of how the receptor can signal even when it's blocked. Because that's another tricky thing, is about a decade ago, a lab found out that the NMDA receptor can actually signal when the magnesium ion is still sitting in the channel and when no calcium is coming in, which was contrary to the last kind of 50 years of research on NMDA receptors. If that answers that.
Yeah, but really quickly, so you said it was sitting on the like the receptor. Is that sort of like a allosteric inhibition or a competitive inhibition?
Oh, that's another really good question. So ketamine, for example, it's just in the same place, is referred to as a non-competitive antagonist. So that means it's going to block the calcium from coming into the cell while it's sitting in that ion channel pore. But kind of the key difference here between these sorts of drugs, so agonists versus antagonists versus allosteric modulators. So you can have positive allosteric modulators or negative allosteric modulators. The difference between an agonist and antagonist is going to be how it binds to kind of the receptor and sometimes specifically what place it's binding to. But with NMDA receptors, at least allosteric modulators, a lot of those actually bind to a different part of the receptor. So you have this kind of clamshell in the receptor that's sitting at the top, and then you have this other one, which is where its neurotransmitter glutamate binds. Below that is where you have the ion channel, which is where the calcium comes into the cell. And then below that, you have what's called the intracellular domain. So MK801 and ketamine are going to be sitting in that ion channel pore where calcium likes to flow in. So if it's sitting in there, then you're blocking the calcium from coming in, and the receptor is not really going to be signaling, as it should, or at least it's, again, a little bit more complicated now that we know that NMDA receptors can signal even when the channel is blocked, but we can get into that more if you guys want in a second. But with allosteric modulators, you can have positive or negative allosteric modulators. And these are going to kind of more specifically change things like the channel kinetics of, that ion channel, for example, how much ions are coming in depending on what type of subunit or activation you're looking at, and a bunch of just different kind of mechanics of the receptor. So to put that a little bit more broadly, when you have some sort of antagonist, that's, generally going to be blocking the receptor from doing something. Whereas when you have some sort of allosteric modulator, whether it be an inhibitor or some sort of activator, it's instead going to be altering how that receptor is working rather than just simply enhancing or blocking it. So if you're interested, there are, quite a few different compounds that work as allosteric modulators with NMDA receptors and affect certain parts of those associated functions like plasticity and learning and memory. One of them is zinc. So zinc acts as an allosteric modulator for NMDA receptors. And then a couple other drugs like that, but they are a little bit different than like ketamine, PCP, MK801, and those that are considered to be just purely antagonists.
So with NMDA receptors and their function and synaptic plasticity, I was wondering if there's like any other like molecular underpinnings of diseases, such as like, do genes and metabolic dysfunction play a role in like altering NMDA receptor plasticity?
Yeah, so another really good question. Obviously, since genes are kind of coding for your proteins are doing all of these things at, or, in different scenarios throughout life. Inherently, if you have some sort of mutation in some sort of protein or kinase, like CAM kinase alpha, for example, that's one of the major kind of proteins associated with that downstream signaling of NMDA receptors that leads to our ability to learn and remember things. If for some reason your gene for CAM kinase is mutated, and people have done this in animals experimentally, then you do see pretty profound alterations in the animal's ability to undergo just normal learning and memory abilities, whether it be kind of behavioral or, while they're navigating a maze and just different stuff like that. So anytime you have some sort of kind of genetic influence over a protein or something like that associated with NMDA receptor activation, you can certainly have some sort of alteration in these functions that the NMDA receptor is associated with. And yeah, and that's another thing that we're kind of studying in our lab a bit as well, is there are a class of genes that are called early immediate genes. And these are genes that are pretty present and pretty active during early development. And one of them is called the ARC gene, or the activity regulated cytoskeleton. And this is one that, again, is pretty closely linked to NMDA receptor activation and the processes that kind of underlie the formation of memory at various kind of developmental stages, but especially when you're kind of really starting to be able to remember and all of that. So that's not my project, but another project we have going on is, as you kind of asked, looking at different types of genes or this type of gene and how changing NMDA receptors or their composition or how they're working affects how that gene is kind of doing its thing in an environment for an animal like a maze and at what developmental stage. So a lot of kind of different effects on that level as well. Especially, again, because NMDA receptors aren't just in the hippocampus, they're all over the brain in different regions, and sometimes they look different based off of where in the brain you are. So given that they're kind of just everywhere and they're playing this really important role in plasticity, even if it's kind of a different plasticity than in the hippocampus, that does kind of make them a bit subject to being changed by or changing certain genetic influences over how they work and how plasticity is induced.
All your research is just so interesting. Like I've just been like intrigued this entire time. Um,
Thank you. I think it's pretty cool too. It's pretty fun to kind of work hands-on with it day to day. So I love being able to talk about it.
yeah, and I'm so glad we were able to give you kind of like a platform to be able to talk about it.
Yeah, me too. And I love the idea of kind of more people learning about this stuff, especially because, a lot of people at this point, even if they're not in psychology or neuroscience, have heard the word neuroplasticity. And it's just purely amazing that the brain has that ability, but it's really complicated and there's a lot that goes into studying it. And it can be kind of hard to study sometimes, but all of that is kind of just what makes it so amazing and so interesting. So I love, when people are interested in that as well, or if I can talk a little bit more about that because I think it deserves a lot of attention. The brain is just amazing. So
Yeah, I definitely share that like passion of like biology and like I know that like right now in like high school there isn't really a lot that you can do when it comes to like going in depth with like just like the inner workings of like everything when it comes to biology. But I remember like my freshman year of high school, I was completely drawn into biology and just how it worked, and I share that similar like amazement that you have. I want to ask a few questions about like um your career so far and everything like that. Is that okay?
yeah, of course.
Yeah, so um I just like want to ask like do you have any like advice for anyone who kind of wants to go into like psychology or just like neuroscience in general?
Yeah, so I would say if you've kind of recognized this spark or this intense interest that you have in how the brain works or how human behavior works, or anything like that, my biggest piece of advice and what I did is to just really kind of enjoy that. And kind of try to facilitate it. As I said, when I was an undergrad, I really liked psychology a lot. But I also really started to like biology, and I wasn't taking a lot of biology classes, but I was really curious. So a lot of my free time, because I was so cool in college, was spent just reading different papers and trying to understand, different scholarly articles or peer-reviewed papers that were talking about experiments that people were doing, with drugs like SSRIs or psychedelics and how those were affecting mood. And just the rabbit holes that I would go down when I kind of just thought of something related to psychology or neuroscience and would then research it. It was really what kind of helped me just really advance my understanding in it. And when I was doing that, it was kind of inherently making me even more and more interested. So I would say to anybody who is interested, just be interested and let yourself be interested. And when you have questions about them in your head, when you're drinking your coffee in the morning and you think of, oh, how could this part of the brain be playing into this? Look it up and read about it, because it's likely that there are some researchers out there who are spending their whole life looking into what you just asked, and there's a lot of good information out there about it. So yeah, that would be my biggest piece of advice, would be to just kind of really foster your interest in it, especially while you have the time to kind of read and like go down rabbit holes, and just be interested in a bunch of different things. Because when you get to your PhD, you can obviously do that, but you also have this very big project that is very niche and very specific. So a lot of your time has to be spent on that. But to kind of give, I guess, more advice that's aimed at the process of pursuing interests, I would say, it's there's a lot to do, especially when you get into college. If you realize that, you want to be a researcher and you want to do your PhD or go to graduate school or something like that, you have to, kind of get good enough grades to get into graduate school. And then a lot of people will want you to have research experience or some sort of relevant experience. So I would say anytime an opportunity like that comes up where you can get some sort of job or work or research experience that might be related to something that you're interested in, psychology or neuroscience, to take it. The best advice I got from somebody when I was in college, after I had really just realized that, I do want to do a PhD in this, was to kind of reach out to people or professors and just ask if they have a spot or need help in their lab. And, you do that with a bunch, and some people will come back and say, yeah, my lab's full. Unfortunately, I don't have a lot of spots left. But some people will come back and they'll say, I'm interested. Let's set up an interview and see if your experience or, your interests align with what I'm doing. So that was my kind of biggest piece of advice that I received from somebody when I knew I wanted to apply to a PhD to go into psychology and neuroscience, was to just reach out and just ask if you can help professors or researchers. And a lot of times they appreciate that, initiative, and they'll talk to you or interview with you, even if your lab is full, and all of that is just really good experience. So yeah, again, to wrap that all up, I would just say, especially when you go to college, if you want to do research, just look at the faculty website, look at whose research is interesting to you, and just reach out to them and ask if they have any help. And we have some high school students that actually do that with some of our labs here as well. So we're busy people in research labs, and even though our labs can get full sometimes, a lot of times, students end up leaving and we need more help again, especially when we have assistants that are like in undergrad, for example. They'll graduate and they'll leave and we'll need more help again. So super important to just keep your eyes open for things that interest you and just kind of shoot your shot with professors or labs or research that you're interested in sometimes.
I love that. Do you guys have anything else to add? Or
Oh no.
No, it's okay. I
all
think
right.
we covered a lot of points.
We did cover a lot, and I thoroughly enjoyed this. This was very enjoyable.
I did as well, and it makes me happy to hear. So thank you guys again for having me.
Of course.
So much for sharing your knowledge.
Of course.
Yeah, thank you so much. You're just a very cool person and is really interested in hearing about your story, and it's just amazing.
Thank you so much. And thanks for asking about my story. I again just had a great time talking with you guys. So
Okay, then. I think we're gonna wrap it up here, just because like this meeting is gonna end, but
okay.
also because we kind of set it like a little time limit for ourselves to just to stay on track. But
Yeah, that's awesome. Yep, it sounds good. And I wish you guys the best of luck on all of your just high school related endeavors, but then everything that you guys want to do or are interested in when it comes to psych and neuro. So,
Thank
yeah,
you.
thank you.
Thank you.
okay, I'm gonna hop off here. Thank you
Yeah,
guys
you're
again.
good. Of
Okay,
course.
bye.
Thank you.
Bye. Thank you.